Hello everyone. Plant breeding aims at the discovery of variation in nature or creation of artificial variation and then delivering useful variants into cultivars. In this way, it plays a significant role in improving crop production. In general, the term plant breeding innovation is used to describe the constantly evolving ideas and practices which enhance the field of plant breeding. In the first part of this video, I'm going to talk about my research story from which you know more or less what challenging issues are in plant breeding. My first round research at the Wageningen University had been done with the topic of potato late blight resistance. The main objective was to uncover the underlying mechanisms of the multi-year persisting late blight resistance in the field, also referred to as durability, in some potato clones and cultivars, and describe how to deploy that resistance. You know, potato is growing in almost all countries of the world and is the most important non-cereal food crop. It produces high yield, has better food quality, can be harvested earlier, and can grow in harsher climate conditions than any other major crop. Like other crops, potato plants are vulnerable to several pests and diseases. The most devastating disease is black blight caused by the omega pathogen Phytophthora infestans. Disease-free seed potatoes, which are produced by tissue culture and mass multiplication systems, and frequent fungicide sprays are effective measures for potato crop protection. However, financial issues for the development of the disease-free seed potato production system are considerable. Furthermore, frequent fungicide applications are harmful to both humans and environment. Moreover, the capacity of the pathogen to develop resistance to modern fungicides, for example, blue 13 and green 33, is a problem for sustainable potato production. Therefore, the use of resistance varieties is the most effective and environmentally friendly approach to late blight management. Also, we are unfortunate to face the reality to break down the resistance of a potato clone in several years after commercialization, even in breeding stages. So we need to develop durable resistance which lasts long since release of a specific resistant variety. Now we have some knowledge about how the plant immune system works. Since the statement of Flow's gene for gene concept involving both plant resistance genes and pathogen virulence factors, the scenarios of plant microbe interactions have been revealed. Here, I show the potato Phytophthora infestans interactions. Several years of our research led to successful mapping of two R genes, and now they position on the chromosome 9 in the Solanacea map. Both of them also cloned successfully. We used the potato differential site clones MAR8 and MAR9 and potato cultivar Sapamira as the plant material for broad spectrum late blight resistance. Through previous studies, we know that among the set of 11 potato gene differentials, MAR8 and MAR9 have shown a broad spectrum resistance in field trials as well as in the laboratory assays using detached leaves. Sapamira was shown to express similar levels of resistance to late blight as the MAR8 and MAR9 in multiple years of field trials in Europe and Northern Africa. In order to test whether broad spectrum resistance is a result of gene stacking or a result of the individual genes, we made F1 and BC1 population and selected plants with combinations of multiple genes. Parents and offspring plants were tested using three approaches, which included agroinfiltration with known AVR genes, detached leaf assays with selected isolates, and gene-specific markers. A remarkable thing was that the test liver-cell results were different from field trial results for PI isolated EPOC in all F1 and BC1 populations. Initially, these results were very embarrassed. We employed a whole plant assay in climate room, and indeed, we obtained reproducible results as the field trial. So it was clear that late blight response to the isolated EPOC works in a whole plant. We found that MAR8 contains at least 4 genes, MAR9 7 genes, and Sapamira 4 genes. A common factor in all these plants was the R8 gene. Analysis of MAR8 and MAR9 of spring plants that contain different combinations of multiple R genes showed that R gene stacking contributed to the PI recognition spectrum. Also, using a PI virulence monitoring system in the field, we observed that stacking of multiple genes strongly delayed the onset of late blight symptoms. The contribution of R8 to this delay was remarkable because plants which contained only the R8 gene still conferred a delay similar to plants with multiple genes like Sapamira. In terms of R8 mapping, I'd like to mention two things. One thing is observation of one-to-one segregation of resistance and susceptibility, which indicates the R gene referred to as R8 is present in simplex in the tetraploid MAR8 clone. And the other thing is in-base profiling as a kind of motive-directed profilings which targets conserved R gene clusters. In-base profiling was employed by using degenerate primers in conserved motifs. Once a particular gene cluster was tagged, primers specific to the gene cluster were used to reach high marker saturation within the cluster. This technique is called cluster-directed profiling, CDP. Through in-base profiling and successful marker sequence comparisons to the potato and tomato sequence genome draft sequences, it was suggested that R8 is located on the long arm of chromosome 9, not on the chromosome 11, as was suggested in a previous study. Analysis of SSR caps scar markers confirmed R8 is on the distal end of the long arm of chromosome 9. Further, CDP markers flanked R8 were developed. One marker, CDP3, was fully co-segregated with the EPOC resistance. Sequential analysis of CDP3 showed that CDP3 had high similarity to the tomato gene SW5. Another remarkable thing in my study was the discovery of AVR8. Based on testing of collection of in-plant-induced RXLI effectors of phytophthora infestans, we screened the effector PITG07558, which showed R8 specific cell death. The response to this effector co-segregated with R8-mediated response to EPOC in F1 population, and therefore this gene was designated AVR8. Interestingly, the response to AVR8 also co-segregated with response to EPOC in Sapomira-derived segregating populations. So we concluded that RPI is MIRA2, which was found by other PhD students in our group, is the same gene as RA. To search the geographical distribution and phylogenetic origin of R8 in the Solanum gene pool, I carried out a functional screen for AVR8 response in 98 wild genotypes of Solanum section petota. From this screening, 12 AVR8 responding Solanum accessions were found, which are originating from Central and South America. As mentioned earlier, MAR9 contains multiple R genes. In order to eliminate the effects of other known R genes, we obtained offspring plants containing different R gene combinations using crossing marker analysis and effector response. It was really essential to discern R8 and R9 clearly because MAR9 contained R8 as well as R9, both of which were resistant to EPO-C. Analysis of the absence of R8 gene in mapping populations was done using R8 marker and in-plant AVR8 expression. Populations lacking R8 marker were further tested by the AVR8 response. To rule out the absence of R8 marker was caused by genetic recombination between R8 gene and the R8 marker. With PCR populations which showed one-to-one segregation of resistance and susceptibility, we first tested other markers near R8 gene and found that the 184-81 marker flanked the R8 gene at 1 centimorgan distance was fully co-segregated with the resistance. The map position of R9 on the distal end of the lower arm of chromosome 9 was confirmed using known PCR markers. Furthermore, CDP was performed and fully co-segregating CDP markers which had high homology to TM2 clusters could be found. A back library derived from the differential plant MAR9 was screened with co-segregating CDP markers based on sequence annotation of the complete insert of the CDP marker containing back clones to complete NBLR type argine analogs RGA 9.1 and RGA 9.2 were identified in one back clone. Two RGAs, including their own native regulatory transcriptional elements, were subcloned by long-range PCR into a binary vector for plant transformation. Complementation analysis showed RGA 9.1 was able to complement the susceptible phenotype in cultivar Desiree. The clone to R9A deduced to encode CC-NBLR protein. In terms of R9A cloning, I'd like to mention CDP approach could produce specific markers landing near the gene of interest. For map-based cloning, the process of targeted saturation with markers for back landing is commonly performed, but is laborious and time-consuming. From our previous work, we experienced that CDP increase the chance to discover co-segregating, even fully co-segregating profiling markers in specific chromosomal regions representing argine clusters. The more co-segregating markers can be found, the higher the chance that back landing of PCR-based allele mining could succeed. Here, we showed that CDP enabled to select back clone carrying the target R gene without the need to perform high-resolution mapping. With respect to cloning of R8 gene, I tried to clone it based on a co-segregating marker position from MAR8 plant-derived back library, as in R9A cloning, but I didn't succeed during my PhD study. Back library screen and recombinant screen were performed, but in complementation analysis, the selected RGAs were not positive. Later on, a new back library was used, which was constructed partial restriction enzyme digestion instead of mechanical shearing that was used in constructing the first back library. Indeed, for additional complete RGAs, 0.10, 0.11, 0.20, and 0.21 were found, and complementation analysis and co-expression of RGAs with AVR8 in The Nicotiana pentamiana revealed only RGA.20 induced HR response. All sequences for 10 parallel RGAs had high homology to the tomato argine SW5. The cloned RA gene also belongs to a typical NBLR type argine. When tested under field conditions, RA transgenic potato plants showed broad-spectrum response to the current late blight populations in the Netherlands, similar to Sapomira. From the aforementioned research, it becomes clear gene stacking would be a promising approach to obtaining durable resistance. We practice the cystogenesis by which native genes from crossable species are introduced into potato varieties using gene technology. In order to bypass biosafety issues associated with transgenic crops, cystogenesis pursues the transfer of native genes with their own regulatory transcriptional elements to the same or crossable plant species. Two broad-spectrum late blight genes, RPI-STO1 and RPI-VNT1, from the crossable species Solanum stoloniferum and Solanum venturi, respectively, which were cloned in our group or introduced into three potato varieties. First, we produced single argine-containing transgenic plants to use as references for the resonance levels and spectra to be expected in the respective genetic background. Then, a construct containing both cystogenic genes, RPI-VNT 1.1 and RPI-STO1, but lacking the bacterial kanamycin resonance selection marker NPT2, was transformed to the varieties using agrobacterium-mediated transformation. Gene transfer events were selected by PCR among shoots which were originally rated on kanamycin-free medium. Shoots were selected which are PCR positive for both RPI genes and also PCR negative for backbone integration. Through further analysis covering morphological evaluations in the greenhouse, AVR gene responses, and late blight resistance in detached leaf cells with 5 isolates, the selection was narrowed down to 8 independent events. These cystogenic events showed broad-spectrum resistance due to the activity of both transferred genes in comparison with single gene introduced transgenic plants. From our study, what we have known and what can be suggested. In this study, we found strong indications the durability of the resistance to late blight mostly associated with the stacking of multiple genes. Durability indicates how long resistance lasts since release of a specific resistant variety or formation of a specific resistant genotype. Phytophthora resistance is able to evolve quickly to overcome gene-mediated resistance. It is much likely that sexual recombination between two mating types increases genetic diversity in PI populations. The question therefore arises whether it is possible to obtain durable gene-mediated resistance, and if so, how can it be realized and maintained? Answers might be found in the nature of our genes or their cognate AVR genes. Our AVR interactions could be categorized into 4 classes. Class 1, single R protein to single AVR protein. Class 2, single R protein to multiple AVR proteins. Class 3, multiple R protein to single AVR protein. And class 4, multiple R proteins to multiple AVR proteins. The copy number variations and abundance of the AVR gene in PI population are major components for producing durability. As a counterpart of such AVR genes, potato genes that show broad spectrum resistance and have probably abundant functional homologs among various wild solanum species could be optional for gene combinations, providing durability. Up to now, an ideal durable gene that is resistant to all PI isolates was not found. Even if such a gene could be available, exposure to strong selection pressure under large-scale agricultural deployment could lead to gene breakdown due to the co-evolutionary predominance of virulent isolates caused by mutation, silencing, or suppression by another effector. For instance, PIBL2 and R9A breaking isolates were found in PI populations of the Netherlands at a low frequency. Also, virulent races on PIBLB1 containing potatoes have been identified in Mexico and the Netherlands. Therefore, the logical idea to combine multiple genes with different spectra would be preferred to achieving durability. Once potentially durable genes are identified, stacking these genes with different resonance spectra, recognizing different PI effectors commonly present in the PI population, would enable to produce appropriate resonance for a long term. With respect to durable gene, R8 is interesting because we found up to 10 functional homologs with AVR8 response among various solanum wild species, and AVR8 gene is present in all thus far sequenced PI isolates. In particular, R8 gene was found in 5 late blight resistant varieties deployed in 3 different continents: Europe, United States, and China. We pursued cisgenesis to obtain gene stacks. Cisgenesis is equivalent to gene technology, and so it allows to introduce multiple genes simultaneously without perturbation of a favorable agricultural genetic background. The process consists of marker-free gene delivery by Agrobacterium pseudomonas and subsequent regeneration in antibiotics-free medium, followed by PCR selection of the stacked gene-containing events. Cisgenesis as a method to bypass biosafety issues was in controversial debates, such as the use of Agrobacterium and plasmid vector and random integration of the transferred genes, etc. In the second part of the video, we can see how to figure out these issues. My second round study at the Wageningen University was to establish CRISPR-Cas9 mediated mutation protocol aiming at functional gene studies. Many experiments have been done with tomato and potato, and interesting results were obtained. My focus was on how to generate mutations simultaneously across all different alleles on homologous chromosomes in polyploid plants. This topic is really required to knock out dominantly inherited genes in polyploid species. Of course, we know that using protoplast transformation or particle bombardment, complete allele disruption mutants in the T0 generation could be obtained after testing hundreds to thousands of regenerate plants. These approaches are laborious and time-consuming and are less suitable for routine use in gene function studies. So, to employ CRISPR-Cas9 as a tool for functional gene studies, I investigated an approach that enables an efficient generation and easy detection of tetraallelic mutants using Agrobacterium-mediated transformation. Generally, when a single sgRNA CRISPR-Cas9 system is used in polyploids, the frequency of knockout mutations is very low. Initially, A single sgRNA construct was used in my experiments to target genes in tetraploid potato digere. As expected, tetra-allelic mutants were not found among the tested 46 Cas9-positive transgenic plants using agrobacterium-mediated transformation. Based on computational work, experimental designs were shifted to the multiple sgRNA system, and indeed, tetra-allelic mutant plants were obtained. It is expected when the number of sgRNAs for targeting the same gene in polyploids is increased, the number of mutation events will also increase. Let's say 1 is mutation event, 0 is no event. For the calculations, I assumed that all sgRNAs have the same activity and each mutation event occurs independently. Each event have two 4-digit sums, namely the sum of rows or the sum of columns. There are sum of events per chromosome and sum of events per target site, respectively. The combinations event ID with one or more 0 in the sum of events per chromosome are not tetra-allelic. In this way, total event combinations can be calculated depending on different numbers of sgRNAs and ploidy. Mono, D, 3, and tetra-allelic mutations can occur, and they include mutations in cis and in trans, or both. Also, among mutations, there would be events with defined excisions between target sites. As you see in the table, theoretical probability of tetra-allelic events increases from 6% for a single sgRNA to 77% for the 4-sgRNA system. These tetra-allelic events include cis-tetra-allelism at the same target sites and trans-tetra-allelism between different target sites. In a separate video, I'll show you how to calculate using Python scripts. You can recognize it is difficult to generate complete allele disruption events by using single sgRNA in tetraploid. By increasing the number of sgRNAs, we can obtain tetra-allelic mutants very efficiently. Importantly, you imagine one-to-one relationship between genotype and phenotype could be present, so the majority of the resulting mutants could display phenotype of interest. I also calculated the probabilities of possible PCR detectable events in different sgRNA systems. They are not shown here. As you know, the simultaneous use of multiple sgRNAs could cause defined excision events between genomic sites targeted by different sgRNAs as an irreversible repair. It is generally thought that the CRISPR-Cas9 has the possibility to employ the PCR approach to detect mutations when a forward primer in the target site is designed carefully. In the multiplex CRISPR-Cas9 application, the forward primer wouldn't bind to the target sites after CRISPR-Cas9 action in two cases. One case is mutation at the specific target site itself, and the other case is the defined deletion between other target sites flanking the specific target site, or between other target sites and the specific target site. In this figure, tetra-allelic mutants can be detected using standard PCR with primers, of which forward primer is designed to be different at 2 or 3 nucleotides upstream of PAM. Here, please note NCC, which is opposite to NGG. In 2 chromosome 1 and 2, there are no sites matching 2 primers because of excisions spanning the target site indicated by dotted line. The nucleotides in red color on chromosome 3 and chromosome 4 indicates in there generated by CRISPR-Cas9. For all four genes, PCR fails, so we observe no wild-type band after agar-gel electrophoresis. The cultivated tetraploid potato variety Desiree was used for demonstrating the multiple sgRNA concept. Vectors containing four sgRNAs were constructed for targeting two potato orthologs and the functional annotation study. We selected two genes because they could be functional orthologs in previous RNA interference experiments. Here in the figure, one gene shows. Also, we set up experiments with the same genes and the same four sgRNA vector in diploid tomato and diploid potatoes. The four non-overlapping target sites were selected across the whole gene, and distances between target sites ranged from hundreds base pairs to thousands base pairs. In experiments, we observed all kinds of the expected size of PCR fragments after a defined excision event. In order to determine wild-type alleles in Desiree, PCR products spanning target sites were subjected to cloning and single sequencing. Sequence analysis of 10 colonies showed relatively highly conserved sequences only with 4 SNPs in Desiree, and 4 wild-type alleles were categorized. This figure shows the PCR-selected tetra-allelic mutants and their mutation profiles. 42 chromatin-resistant transformants of Desiree were analyzed for tetra-allelic events. 4 plants, 4p13, 4p21, 4p31, and 4p37 were selected as tetra-allelic mutants because the wild-type bands in PCR at target sites were absent. Sequences for the used primers are conserved in all 4 wild-type alleles, so that it strongly indicated the 4 alleles in the mutants without PCR product were completely disrupted. The PCR product covering the mutated target site was cloned. The cloned fragment also didn't have wild-type band in PCR products using the primers which were used to select tetra-allelic transgenic plants. In the figure shows mutation profiles of the PCR amplification for the target site 2 in mutation plant for P13 and for P21. 205 base pairs of small deletions occurred in the target site for P13 mutant plants and deduced to cause 3 consecutive stop codons downstream of 7 amino acids from target site in the predictive protein sequence of the gene. For mutant plant 4p21, the 65 base pair deletion occurred and resulted for amino acid change as well as 3 consecutive stop codons for the predictive protein sequence. It turned out that mutations for the other wild-type alleles that couldn't be detected from sequencing analysis came from excision events between target site because we observed the expected size of fragments in follow-up experiments. The arrows in figure indicate natural SNPs that exist in wild-type Desiree. Now I'm going to wrap up my CRISPR-Cas9 study. In this study, agrobacterium-mediated CRISPR-Cas9 induced mutagenesis was investigated. Based on computational work, the 4 sgRNA CRISPR-Cas9 system was tested on diploid tetraploid levels. It could induce a highly increased number of biallelic tetraallelic events in the T0 generation. The majority of the resulting transgenic plants display mutant phenotype, so that if needed, molecular detection using standard PCR could be employed only in young plant stage. We also tested another four sgRNA construct in which two sgRNA were overlapping. We didn't get similar or better results compared to the non-overlapping 4 sgRNA system. Therefore, when the 4 sgRNA are selected, non-overlapping ones could be preferred. We observed the various sides of expected excisions as well as some unexpected excision events. As one reason of unexpected excision, multi-copy integrated tDNA could cause unexpected excision events. It remains unknown how such fragments are occurring. The important thing in my study was that we couldn't get the similar results as the RNA interference results. We used the same 4 sgRNA vectors targeting the same target site sequences for diploid tomato, diploid potato, and tetraploid potato. Unfortunately, the expected mutant phenotype that was observed in RNA interference experiments in tetraploid potato couldn't be observed in my experiments. So this study is one example showing discrepancy between CRISPR-Cas9 and RNA interference. It will require careful considerations for RNA interference experiments. In particular, in polyploid organisms which contain multi-copy number genes, the computational annotation of functional homolog or orthology should be validated using advanced mutagenesis tools like CRISPR-Cas9. Since plant domestication started about 10,000 years ago, plant improvement has depended upon selecting the best performing plant in the field. The discovery of Mendel's law of heredity in 1865 has opened the door to develop improved varieties by combining good characteristics from two parents. This crossbreeding has been supplemented with hybrid breeding, which cross two genetically different individuals to develop better performing hybrids, and mutation breeding, which develops new genetic diversity by exposing crop plants to chemical agents or radiation. After the discovery of DNA structure, plant breeding has utilized genetic information to develop improved varieties. The use of biotechnological tools like genetic transformation and macrocystic selection has facilitated breeding processes in more accurate way and with a short time span. With coming out efficient gene editing approach, we are fortunate to leave the era of precision plant breeding, which holds great promise for improving crop varieties. The CRISPR-Cas9 system derived from bacterial clustered regularly interspaced to the short palindromic repeats CRISPR-associated 9 protein Cas9. CRISPR-Cas9 system is a rapidly developing genome editing technique because of simplicity and robustness for specific removal, replacement, or addition of DNA sequences compared to other sequence-specific nucleases, such as zinc finger nucleases and transcription activation like effector nucleases called talons. The standard CRISPR system used for target mutagenesis is composed of two parts, Cas9 endonucleases and a single guide RNA molecule. Cas9 identifies the target DNA by base pairing between the target and the sgRNA. For Streptococcus pyogenes Cas9, which has been widely used, The proto-space adjacent motif, PAM, is 3-nucleotide NGG. PAM is required for binding to the target DNA sequence. Upon binding of the target DNA by the Cas9 sgRNA complex, Cas9 initiates double-stranded breaks, DSB, between the third and fourth nucleotides 5' to the PAM. Repair of the DSB by the error-prone non-homologous end joining and HEG pathway typically results in short insertion and a deletion that can produce knockout mutations in protein coding sequences. When a template with regions of homology to sequences surrounding the DSB is available, the DNA damage can be repaired by homology-directed repair HDR, and this mechanism can be exploited to achieve precise gene modification or gene insertion. Unlike DSB-mediated genome editing, base editing systems that can induce specific base changes do not depend on HDR or donor DNA and do not involve the formation of double-stranded breaks. The cytidine base editor CBE system, which consists of a cytidine deaminase fused with an N-Cas9, also called DNA nickase, and a uracil glucosylase inhibitor, converts targeted histogene to thymine in genomic DNA. Adenine base editor ABE mediates the conversion of adenine to guanine in genomic DNA. Base editing system can be used to efficiently create the point mutation in the target site. The new technology, called integrate, harnesses bacterial jumping genes to reliably insert any DNA sequence into any genomic site without cutting DNA. Integrate stands for insertion of transposable elements by guide RNA associated targeting. The site where the enzyme integrates inserts the DNA is completely controlled by its associated CRISPR system. The technology is expected to be tested in plant cells as well. Plant breeding covers 3 different sections: discovery of variations in nature, creation of artificial variation, and delivery of useful variation into cultivars. Why CRISPR-Cas technology is representative for precision breeding? What are unique properties of CRISPR-Cas technology? In terms of discovering variations, traditional breeding requires many lengthy and tedious processes, including crossing, mutation, mapping, and cloning. In contrast, precision breeding requires targeted mutation followed by direct cloning. To create new variations, traditional breeding is undertaken by hybridization between 2 different genotypes and also by mutation breeding. The nature of mutation is random and undirected, while in precision breeding, mutation is highly precise and directed. Once a specific gene or gene candidates are defined based on sequence information and bioinformatics, loss of function or gain of function mutations can be obtained in 1 generation. In contrast to traditional mutation breeding, simultaneous mutations at multiple alleles can be created in polyploid plants. Once useful variants are defined, traditional breeding undertakes lengthy and tedious crossing and intentional selections. Using biotechnological tools like Markov useful selection or genetic transformation, speed and accuracy of breeding could be improved. Tedious backcrossing in Marcos selection and regulatory issues in gene technology are still limitations to wide use of these molecular breeding tools. In precision breeding, the desired traits are produced by delivering RNP into elite cultivars. Also, haploid induction edit can be used, which enables direct editing of elite lines by a single cross. The multiplexing ability of CRISPR-Cas9 technology enables immediate pyramiding of multiple beneficial traits into an elite background within one generation. Considering 2 or 3 years field trials, it takes 4 or 5 years to deliver a germ-free edited variety. In terms of speeding up breeding, de novo domestication of wild species is interesting. 2 independent studies have been published in which accelerated domestication of wild tomato extensions has been demonstrated. By simultaneously editing for genes involved in plant architecture SP, flowering time SP5G, and fruit size CLV3 and US, domesticated tomato has been obtained. Cas9-free progeny of edited plants had domesticated phenotypes but retained parental disease resistance and salt tolerance. This shows that by targeting so-called domestication genes with CRISPR, the domestication process can be accelerated dramatically, and it could be highly useful to open up the enormous genetic diversity present in wild species. From domestication experiments in tomato, we can imagine domestication of brassica wild species within one generation could be achieved using multiplex CRISPR-Cas9. Over the last few thousand years, farmers have been bred brassica oleracea into 6 common vegetables that many people buy and eat on a regular basis. If domestication of this wild mustard plant are demonstrated, we could have very exciting new opportunities about the development of brassica varieties. In this article, I'd like to mention 2 topics using CRISPR-Cas9. 1. How to fix heterogenesis of F1 hybridized seeds, and 2. How to obtain edited seeds lacking both haploid-induced parental DNA and editing machinery. Current hybrid rice seed production is still labor-intensive, and price of hybrid seeds is very expensive. Chinese scientists demonstrate the possibility to get clonal hybrid seeds by combining fixation of heterozygosity and haploid induction using simultaneous editing of 4 genes in hybrid rice. Combined mutation of 3 genes OSD1, PAIR1, and REC8, which mediate crucial meiotic processes, create a genotype in which meiosis is replaced by a mitosis-like division. So male and female clonal diploid gametes are produced. However, self-fertilization of this plant doubles the ploidy at each generation. When this plant is crossed with a SENA3 mediated chromosome elimination line, diploid offspring is produced clonally. The simultaneous editing of all 4 genes in hybrid rice resulted in plants which could propagate clonally through seeds. The MTR gene used to induce parental genome elimination had negative effects on hybrid fertility, and so modifying the MTR gene or looking for different haploid-inducing genes is still required. Haploid induction edit, HI edit, enables direct editing of elite inbred lines by a single cross. This technology utilizes haploid inducer line carrying a CRISPR-Cas9 cassette targeting for desired agronomic traits to pollinate elite lines and to generate edited haploids in the elite background. The edited haploids are Cas9-free due to post-fertilization genome loss and could be doubled spontaneously or artificially to generate edited doubled haploid line with improved traits. At present, whole genome sequencing has been completed in several major crops such as rice, potato, and tomato, and so on. So, major goal of the research for the crops has been the functional characterization of all annotated genetic loci involving various biological processes. Unlike conventional mutagenesis libraries in plants derived from fast neutron EMS, TILings, and transposon or tDNA tagging approaches, the ability CRISPR-Cas system to make targeted knockout lines provides a particularly powerful alternative for plant functional genomics studies. Genome-wide CRISPR-Cas9 libraries are routinely used in animal cell culture systems. Several genome-scale CRISPR-Cas9 mutagenesis systems have been established for mammalian cells using array-based synthesized sgRNA libraries, allowing effective genome-scale loss-of-function genetic screen. For plants, libraries of targeted knockout mutants using CRISPR-Cas9 have been reported in rice and tomato. Here you can see how to construct a genome-wide targeted mutants library in rice. First, sgRNA oligos are computationally designed to satisfy stringent conditions. For example, the 12 base pairs sequence matches only one site in the genome. sgRNAs targeting constitutive exonic coding sequences near the start codon are selected, and GC content and secondary structures of sgRNA are considered, and so on. In addition, 20 and 25 base pairs sequences to both ends of the 20 base pair guide sequences for downstream PCR and ligation. Second, construction of plasmid library. Oligos are synthesized by array-based oligonucleotide pool synthesis. Oligos are amplified by PCR and the oligo pool is cloned into the CRISPR-Cas9 binary vector. The accuracy and coverage of the pooled plasmid library are tested by single sequencing and deep sequence respectively. Also, the accuracy and coverage of the sgRNA library in Agrobacterium are tested. The recipient plant, which can be efficiently transformed, has a good fertility and has a fully sequenced genome, is selected and infected by Agrobacterium tumefaciens. Independent stable transgenic T0 plants are obtained, and the quality and coverage of the resulting mutant library are evaluated. Furthermore, off-target effects of the mutant lines are tested. In traditional mutant libraries, the relationship between genotype and phenotype is usually very weak, and it is difficult to identify and clone the genes corresponding to particular phenotypes. It was shown that genotype and phenotype are much more closely related in a large-scale CRISPR-Cas9 knockout mutant rice library than in previous mutant resources. In terms of construction of such mutant library in polyploid crops, we can use the multiple sgRNA for targeting each candidate gene. For example, tetraploid potato can be infected by the pooled Agrobacterium harboring the for sgRNA containing CRISPR-Cas9 expression cassette. As shown in my CRISPR-Cas9 study, the majority of transgenic potatoes could display phenotypes of interest in the T0 generation. Then the targeted gene can be easily cloned with the guide sequence. As an innovative gene discovery and cloning method, there is an approach by sequence capture and association genetics, which referred to as AG-RNSeq association genetics as an enrichment sequencing. The approach came from an alignment-free method for KMO-based association genetics of categorical phenotypes in human disease and bacterial antibiotic resistance. And it identifies, unlike GWAS, associations with structural variations and sites not present in the reference genome. A genetically diverse panel of accessions is phenotyped with different pathogen races and subject to RNSeq, followed by assembly of the NLR repertoire and extraction of NLR k-mers for each accession. RNSeq analysis is undertaken on the diversity panel using the sequence capture-based library and Illumina short-read sequencing with paired end sequence obtained post-accession. De novo sequence assemblies were analyzed with NLR parser software, which identifies NLRs based on number and order of short conserved NLR sequence motifs. The KMO sequence are given a numerical value based on the resistance level, and these values are summed up for each KMO. The AG-RNSeq procedure directly identifies the function of NLR that confers resistance rather than identifying a gene cluster for subsequent candidate gene confirmation. It is also not restricted to the limited genetic variation and recombination present in biparental populations. Additional crossing or mutagenesis is not required for gene cloning using this approach. It requires only phenotyping of an enrichment sequence to diversity panel. Current cisgenesis remains to address critical arguments by which cisgenic plants are not distinct in transgenic plants due to the technology used, such as Agrobacterium tumefacciens for transformation. CRISPR-Cas9 mediated deletion of the targeted genomic fragments followed by targeted integration of cisgenes have a great promise in terms of practical agricultural deployment of the cloned useful genes, which are still in stock. For example, cisgene knock-in, which is elimination of the GBSS1 genomic fragment followed by HDR-mediated or probably integrate-mediated targeted integration of broad-spectrum resonance gene as a cisgene provides significant contributions to both basic research and applied research for development of new commercial potato varieties with amylose-free starch and resistance to late blight. The breeding process using the CRISPR-Cas9 ribonucleoprotein delivery system constitutes first, design of active sgRNAs, second, preparation and intracellular delivery of the cargoes, which are cell penetrable and non-plasmid, third, plant regeneration, and finally, selection of the desired phenotype. At the moment, protoplast or particle bombardment mediated delivery of cargoes are reportedly successful. More easily applicable and genotype-independent delivery platforms, for example, infiltration of cargoes into leaves or shoot apex, or stem cuttings could be required. To facilitate movement of cargoes into cells with cell walls, penetrating peptides could be used. Field-based phenotyping is a critical component of crop improvement because any phenotype is the ultimate expression of the relative effects of genetic factors, environmental factors, and their interactions. Although high-throughput phenotyping platforms enable the capture of detailed non-invasive information throughout the plant life cycle, the results from controlled environments are distinct from the actual situation in the field. With rapid development in the field of image capture, image processing softwares, and drone technology, drone-based field phenotyping comes out as a promising alternative. It enables rapid and non-destructive acquisition of the phenotypic information of crops in the field in terms of accelerating the efficiency of large-scale crop breeding. The traditional methods for measuring crop traits such as biomass, leaf air index, and yield depend on manual sampling, which is time-consuming, inefficient, and inaccurate. There are definite advantages for field-based crop phenotyping using drones, which include high technical efficiency, low cost, stability for complex field environments, timely field work, high resolution data acquisition, high operating efficiency, etc. For example, MapHair is an image processing solution for drone-based phenotyping, which was developed by FITA, an independent European research and technology organization. It allows plant breeders and researchers to carry out high-resolution, high-throughput phenotyping on experimental fields of any size. Still, there exist some limiting factors such as the low capacity of drones, the strict airspace regulations, the lack of method for fast data processing and models for estimating complex traits under different environmental conditions.